So welcome back, everyone, to GCI Research Rounds. Stephanie has her convocation today, so we all wish her a happy graduation. So I'm going to be hosting this rounds. So she sent me a script, so I'm going to go through this right now. So before we get started, I'd like to take some time to acknowledge the traditional and ceded territories that we're each joining from today. For myself, I'm joining today from the territory of the Coast Salish peoples, and those are the territories of the Musqueam, Squamish, and Tsleil-Waututh nations. So next we'll jump into some brief housekeeping. This is just a reminder to everyone to please make sure your microphones are muted for the duration of the presentation. Please feel free to use the chat function for any questions you might have throughout the talk, and we'll have a Q&A session at the very end. So this meeting is being recorded and will be made available on our website. So before I ask Dr. Carey and his lab to start, I just wanted to give a quick reminder to everyone that we're currently accepting LOIs for the GCI CTG Accelerating Grants Program until May 11th. So if you have some research ideas for that, please feel free to submit an LOI, and it will be reviewed for fit for this competition. So, before everyone goes ahead and puts in a full application, we will review it for eligibility and fit. So once again, thank you everyone for joining today. So Dr. Mark Carey, I'm very pleased to introduce Dr. Mark Carey, who's a gyne-oncologist, and his laboratory is focused on tumor research models, especially low-grade serous carcinomas, as well as tumor molecular characterization and preclinical drug testing of several gyne cancers. The ultimate goal of his research is to identify better drug therapies and predictive biomarkers for these cancers. So today, joining Dr. Mark Carey will be members of his team, including Dr. Nelson Wong, Dr. Marta Eduardo Fernandez, and Madison Bittner. So Dr. Carey, I'll pass it over to you now.
Thanks very much, Michelle. Thanks everyone for joining. Can you hear me all right? Great. I'll presume that's a yes, but I was just reminded, I think my wife was trying to join the presentation, so that Maddie told me I should let her in. So, so hopefully she's joining us now. Well, welcome everybody. It's a pleasure to share with you today a little bit about what we're working on and some of the things that we're excited about. And I thought it would be best actually to have some of the wonderful people I get to work with on a day-to-day basis share some of their work with you and share some of the topics of interest. I do recall, so we're going to talk a little bit, we'll give you a little smattering of some of the work we're doing. And to get this all started, if I can control the slides here. All right, now, right. So actually, I remember as a resident, this was quite some time ago, when I was doing my residency at Western, I remember vividly standing outside of, standing at one of the nursing stations with one of my consultants. His name was Bob Casper, and he is a reproductive endocrinologist. And I remember we were discussing a patient problem. And obviously there was absolutely no evidence or guidance to direct us as to how to manage the problem. And I remember distinctly him saying, "Oh, I think we can figure that out in the lab." So away he went to figure it out. And I noticed with many of the things that he was able to do is he took questions that were relevant and pertinent to patient care right back into his laboratory. And I mean, this is nothing new. Everybody's aware of that, trying to create the relevance for the research we do. And the nice thing about research is sometimes even when we don't know that the work we're doing is relevant, it becomes relevant. So part of it is trying to keep our eyes open for what other work people are doing that we can create that relevance for patients. So, it's a challenge because there's many things to study, and cancer is a complex disease. So, but that's the one of the exciting things that we're able to do, and that we look to do with the group and some of the projects that we're working on. Now, we're going to talk a little bit about biomarkers today and share with you some of the work we're doing in this regard. But this is really one of the key elements of the work that needs to be done in the future, not only in terms of what we call biomarkers when we talk about the molecular classification of diseases now. I mean, essentially they're biomarkers because every single cancer is different. So it's really important that we figure out ways of distinguishing cancers from a therapeutic perspective so that we can select the right treatments for them. And now that some of these treatments have become, so much more expensive, we have to actually figure out biomarkers not only to identify the molecular subtype, but we also have to figure out which predictive biomarkers are going to help us select treatments that are costly. And this is a little recent publication there was a comment in this about a large study that was done from 2010 or 2000 to 2010, I think it was, in the United States. But they showed that about 40% of retirees, there were almost 10 million retirees surveyed, but about 40% of them expended their life savings within 2 years of a cancer diagnosis. And so these drugs have become very expensive. And so it's really important for us to develop predictive biomarkers to select patients that will benefit from the drug, limit drug toxicity and needless visits to hospitals when they're not benefiting from the treatment, time away from their families, but also the issue of financial toxicity, because this is a real issue for us. It's somewhat hidden in our system because we don't actually, nobody tells us how much we're spending on a new drug, but some of the new drugs that we use on a routine basis now, run for anywhere from $10,000 to $13,000 a month. Oh, is that it? Yeah, great. So, I mean, we don't have to go over this. I think, this is work that the OCare team has done here in terms of highlighting the importance of the molecular classification and the different subgroups of ovarian cancer. And this has been groundbreaking work done by the OCare group here at UBC. I think we're all familiar with this, that the histology only tells a small part of the story, and we're now appreciating the different subgroups of cancers, even within our own specific histotype. Now there's subgroups of high-grade serous cancers, and now even subgroups of low-grade cancers. And these all have implications in terms of screening, treatment, and clinical trial design as well. So within the low-grade, these low-grade serous cancers that we're interested in studying, and we'll highlight that a little bit later. I think many people are familiar with this, but the fact that these low-grade cancers are more indolent, tend to occur in younger women, and there's been much less research on low-grade serous cancers, so we have many fewer treatment options. But we're now starting to appreciate from the work that's been done that there's different molecular subtypes of low-grade serous cancers. There's some that express ER/PR, estrogen receptor, progesterone receptor. There's some that have 9p loss, chromosome 9p loss, leading to loss of P16 and P15, which are cell cycle regulators. And then there's a group of tumors that have USP9X mutations. And then also there's this emerging no specific molecular profile as well. So there are probably going to be different treatments for each of these subtypes. And it really starts with one thing that we've really been fortunate to have is the support of our patients. And partly it's because funding is difficult to access, but this is Sarah McKenzie and her family, and she unfortunately passed away from low-grade serous cancer about 3 years ago. These are her in-laws as well, the Lawlers, but they've been really big proponents of our research initiative, participating in helping with fundraising and been key in terms of our development as a program. So it's been nice to really link in with patients. We've had other patients that have participated in committees and specific research projects as well. And then we've been able to link with other groups that are developing international collaborations for the study of low-grade serous cancer. This is Dr. David Gershenson, who's one of the world authorities on the disease. And then Jane Ludeman here on the left, she has a low-grade serous cancer and has started a large fundraising program that now she's rolling out through across as many countries as possible to facilitate collaboration in the study and treatment of the disease. So just to highlight this, and we'll go into this a little bit later, but to highlight, there's really been 3 main treatments for low-grade serous cancer, and because it's ER/PR positive, there's been a focus on anti-hormone therapies, and there actually is not a lot that's really understood about this. The standard treatment's always been platinum-based chemotherapy, but there's a limited response. Even in primary treatment, the response rates maybe run about 30%, so it's very different from high-grade serous cancer. And then recently, with targeted therapies, because these cancers have MAP kinase pathway alterations, there's a recent study in relapse disease showing that MEK inhibitors have efficacy better than chemotherapy in the treatment of relapse disease. So we have our work cut out for us, because even in the relapse setting, about 75% of patients aren't really benefiting tremendously from this treatment, and MEK inhibitors have significant side effects. So with that, I'd like to introduce a group of very talented individuals that I have a chance to work with on a daily basis. Nelson's going to talk a little bit about the work he's doing on anti-hormone efficacy in low-grade serous cancer. Maddie's actually working on similar aspect of treatment, in that she's working on ER/PR treatments for breast cancer and cell cycle inhibitors in breast cancer, but this treatment also relates to the treatment of low-grade serous cancer. And Marta's going to talk a little bit about the work that we're doing on combination therapy with dual MEK Pan-RAF inhibition in low-grade serous cancer. So with that, I'll turn you over to Nelson, I guess, right? Thanks, Nelson.
Thank you. So I hope everyone can hear me okay. So as Mark mentioned, one of the major research focus in the lab is to study low-grade serous ovarian cancer, or LGSOC. So as you may know, this is a rare ovarian cancer, and it's usually diagnosed with younger women, and it is rather understudied. This cancer is known to be less responsive to chemotherapy, but there are certain molecular features of this cancer that may help us to look for better management. For example, these cancers are generally express ER and PR, and there are certain proportion of these cancers also have alterations with the MAP kinase pathway, making them susceptible to something like MEK inhibitor. And they generally has wild type P53, and it's very rare that you would see BRCA mutations with these cancers. And also, as Mark mentioned, there's generally, it's quite common that there's a MEKi loss with this cancer. So to study these rare cancers, we need models. So in the past, the lab has actually established a large panel of low-grade serous carcinoma cell line. But to complement the model that we have already established, we set out to also establish patient-derived xenograft models, hoping to better capture certain biology of this cancer. So with this cancer, we chose to do subrenal capsule implant. So it's actually shown here. I hope you can see the pointer. Oh, I don't think
You could just use that. Yeah. No.
So with this implant method, we basically just put the tumor tissue under the renal capsule as shown here. This method is known to be able to better establish patient-derived xenograft models of PDX with not as aggressive cancers, for example, hormone-naive prostate cancers. So we get consented patient samples, and then we'll put them into 4 to 8 mice, and then both kidneys will receive the implants, and then we'll frequently monitor the mice for up to a year, and then at that time the mice will get relatively old, so the mice will be euthanized by that time. And sometimes we don't have any growth, and we'll consider those did not engraft. And sometimes there'll be minimum growth, but the tissue looks viable, then we'll do something that's called host change. But if we observe growth of the tumor, they will be regrafted into another series of mice, and then we'll call that the second generation. And then we'll keep doing that until 4th to 5th generation. Then we'll consider the line established. So we'll consider though that's something that we'll call transplantable PDX lines. And then after the 3rd generation, when the PDX can grow pretty well under the renal capsule, then we will also assess them and see whether the tumor can also grow subcutaneously. And during the process, we collected all the samples for histology and IHC characterization, for sequencing and potentially proteomics analysis. And we do save some tumor seeds that we can recover the models in the future. So in the past, we were able to implant a series of consented patient samples. And all of them are actually from recurrent LGSOC cases. So just to show you some of the features that we're able to capture with our PDX models. So this is one example. So this with HNE, that's the histology. So we can actually preserve the histology pretty well. And it's, in this case, it's stain positive for WT1 and PAX8. And then the P53 actually show wild type staining pattern. And then in this case, we're able to capture the ER expression of this sample, which is something that we are not able to do with the cell culture system. So another thing, this is another example that we're actually able to have the growth of the PDX under the renal capsule. But in this case, this PDX can also grow subcutaneously, and it retains the histological features of the PDX. And then when it grows, this is what it looks like. There's a long lag phase after the implant, but then when they grow, then the doubling time is about 8 to 10 days for this particular model. And there are also, this is an example of a PDX model that actually shows some invasive feature. I hope you can see this on the left panel. That's what you're seeing here is actually the kidney tubules of the mice, and there's some glomeruli, and on the right-hand side is actually the tumor tissue. I hope you can see that there's some invasive feature of the tumor. It's invading into the kidney parenchyma. And then also in this particular model, the tumor is actually metastatic, so it actually goes to the lung. And I also hope that you can see this nodule and also preserve some histological feature of the PDX. So far we're able to have a few of these models engrafted, and then 4 of them have been developed into transplantable tumor lines. So we can consistently use these lines for our experiments. And as you may agree, so with these PDX, we're able to capture certain unique features, and we hope to use these unique features captured by the PDX to address some clinical questions. And recently we have received funding to look for biomarkers that can predict responses to anti-estrogen therapy in a low-grade serous carcinoma. And as you may know, so ER expression is very frequent in low-grade cancer. These patients, most of the time, they will have anti-estrogen therapy, but the response rate is very low. So our goal is to find the biomarkers and then to avoid over-treatment of these women. And also in the process, you'll also lower the cost of healthcare. So this is just a simple schematic diagram of what we plan to do. So we will have the low-grade PDX models implanted into mice. They will either be subjected to estrogen supplemented condition or estrogen deprived conditions. So with the estrogen deprived condition, the mice will undergo ovarectomy. And then after a period of time, we will collect the PDX tissue, extract the RNA, and then perform RNA sequencing. With that, then our goal is to get a set of ER responsive genes. So in the process, we will compare the gene expressions between these two conditions in the ER positive PDX model. We will also do the same thing with the ER negative model as a negative control to remove some non-specific genes. And we will also look into some of the known ER responsive genes in breast cancer. So once we've got a gene set of ER responsive genes, then we will customize the gene set using a nanostring platform. And then using that, we will assess the expression of this set of genes with LGSOC cases of known anti-estrogen therapy response. So from that, we will actually derive a set of genes that should be predictive of who actually would benefit from anti-estrogen therapy and who would not. And then after that, then of course, we need to validate the gene set. So up to this point, we have implanted 14 samples from 13 individuals. With the technique that I mentioned, we're able to have an engraftment rate of about 55%. And 4 of these engrafted PDX has been developed into transplantable tumor lines, and they do retain the histological features of LGSOC. And 2 of these lines are useful in the sense that they can grow subcutaneously, so make future experiments more easier. And then the future plan is we'll continue to characterize these models. And as mentioned, we'll use these models for functional studies, including looking for biomarkers for anti-estrogen therapy. And we do hope to continue to develop more of these models to better capture the tumor heterogeneity. Because as Mark mentioned, even within low-grade cancers, there could be different molecular features. And then we also hope to develop in vitro systems from the PDX models for higher throughput drug screening. So with that, then I'll just hand over the time to Maddie.
Great, thanks. I think we'll take questions at the end if that's okay, unless there's some burning questions for Nelson. It's okay. I don't know if I can see people's hands up here, but maybe we'll take them at the end if that's okay, Michelle. So with that, I'd like to introduce Maddie. Maddie's our master's student, and she's doing some really interesting work on a drug that's being used both in breast cancer and low-grade serous cancers. So Maddie.
All right, thank you for the introduction. So I'll be presenting on targeting the cell cycle in estrogen receptor positive cancers. So as everyone knows, our main focus in the lab is on low-grade serous ovarian cancer. But recently we also have become interested in evaluating breast cancer as it shares some molecular features with low-grade. So from these cancer statistics published last year, you can see that breast cancer is the most commonly diagnosed cancer, and it accounts for 1/4 of all new cases. So ovarian cancer affects not quite as many women. It accounts for 2.7% of new cases. But when you look at the mortality rate of ovarian cancer, it moves up into the top 5 cancer-related deaths. So both breast cancer and low-grade are hormone-driven cancers, with 80% of breast cancers and 50 to 90% of low-grade cancers expressing the estrogen receptor. And so treatment options for both of these cancers include anti-hormone therapy with varying degrees of response. And so another common feature of these cancers is that the cell cycle pathway is altered in a large number of patients. And so in the last decade, there have been novel cell cycle inhibitors that have been developed that specifically target this deregulated pathway. So the mechanism of these cell cycle inhibitors shown on the left here. So P16, as previously mentioned, is commonly lost in low grade as well as breast cancer. So this is a natural cell cycle inhibitor. And so the loss of this protein leads to uncontrolled proliferation from the G1 to the S phase of the cell cycle. And so cyclin-dependent kinase 4/6 inhibitors have been developed, and that mimics the P16 function. And so, as most of you guys know, combination therapies have demonstrated increased patient response rates compared to single drug treatments. And so for this project, we're evaluating different drug combinations using a CDK4/6 inhibitor in both breast and low-grade cancers. So in low-grade, mutations within the MAP kinase pathway are common and have become the standard of care for these women. And so the proposed drug combination is a MEK inhibitor in combination with a CDK4/6 inhibitor. And then in the breast cancer, due to higher rates of anti-hormone therapy response, the proposed combination is tamoxifen in combination with a CDK4/6 inhibitor. And so why are we interested in looking at these specific combinations? So in low grade, there's limited preclinical work done with these cell cycle inhibitors, but there are 2 trials that are currently recruiting low-grade patients to test the combination of a cell cycle inhibitor with an anti-hormone therapy. And then in breast cancer, there's been multiple clinical trials that have actually led to the approval and a change in standard of care practice for advanced disease. So the Paloma-1 trials evaluated the efficacy of Palbociclib, a CDK4/6 inhibitor, in combination with different anti-hormone therapies. And while the results were promising with an increased progression-free survival and overall survival, there were still 45 to 75% of patients in the combination groups who did not respond to this treatment. So this highlights the need to better select which patients are more likely to respond to this treatment. So because CDK4/6 inhibitors are now being evaluated in low grade and have become the standard of care in advanced breast cancer, there's a need for predictive biomarkers to ensure that the right drug is being given to the right patient, while preserving the quality of life for the patients who are unlikely to respond, as well as limit the financial toxicity that comes with targeted therapies. So novel drugs are very expensive, and Palbociclib alone cost the BC healthcare system over $9 million in 2020. So the aims of this project are to evaluate response to Palbociclib in combination with targeted therapies in both breast cancer and low grade, as well as identify predictive proteomic biomarkers of response to the CDK4/6 inhibitors. So to briefly go over the methods, we did in vitro drug testing using live cell imaging and cell counting as the endpoint. We also did some proteomics using reverse phase protein array, and then some Western blotting for biomarker evaluation and validation. So getting into some of the results, Palbociclib was tested in combination with tamoxifen in a panel of breast cancer cell lines. And a range of responses were seen across the lines. And so there were some lines shown in the middle here that only responded to one drug. So SKBR3 only had a response to Palbociclib. Other lines such as MCF7 responded to both drugs, and also the combination showed synergistic results. And then finally, there's also lines that were completely resistant to both of the drugs. So from these cell counting experiments, the breast cancer lines were ranked based on their response to PLD. and they were grouped into responding lines and non-responding lines. So next we looked at the expression of common cell cycle proteins by Western blot. And we found that the two lines that were completely resistant to Palbociclib treatment had loss of RB expression and overexpression of P16. And then from our RPPA data, we did a non-parametric test and we found that there is 13 significant hits identified between the responsive cell lines and the non-responsive cell lines. And so from these candidates, we selected cyclin D3 for further validation, as it is directly involved in the cell cycle pathway. And it was confirmed by Western blot that increased expression of cyclin D3 was correlated with the responsive cell lines. So next we evaluated Palbociclib response in the low-grade cell lines in combination with trametinib, a MEK inhibitor. So we used serum doses to better reflect the level of the drug that's seen in the patients when they're treated with these drugs. And so the serum dose of trametinib is 50 nanomolar, and for Palbociclib, it's 250 nanomolar. And so trametinib demonstrated greater efficacy compared to Palbociclib alone in both of the lines shown here. But there was synergy seen with the combination. And it was actually found that reducing the trametinib dose by 10 times and combining it with half of the serum level dose of Palbociclib elicited the same response as trametinib alone. So this highlights the combination may be beneficial in reducing the toxic effects that are commonly associated with trametinib. And this combination is being evaluated in the remaining cell lines. So we also looked at the common cell cycle markers in the low-grade lines. And it was found that these lines are resistant, or sorry, the lines that are resistant to MEK inhibitor actually had a more intact cell cycle pathway with higher RB expression and higher CDK6 expression, suggesting that this line or this group of lines may derive increased benefit when treated with the cell cycle inhibitor. And so in conclusion, the breast cancer cell lines, Palbociclib had a cytostatic response in 90% of the lines. Loss of RB and high levels of P16 were correlated with Palbociclib resistance, and cyclin D3 expression was increased in the responsive lines. And then in the low-grade lines, Palbociclib had inferior inhibitory activity than trametinib, but the combination was synergistic, and we found that we could decrease the trametinib dose when used in combination with Palbociclib. And then, sorry, just moving forward, we plan to further validate the potential biomarkers of Palbociclib in the breast cancer lines, as well as look into any combination or biomarkers of the combination treatment, and finally biomarkers of Palbociclib plus MEK inhibitor in the low-grade lines.
Thanks, Maddie. I might just point out, this has been a huge, Palbociclib and the CDK4/6 inhibitors has been a huge advance in the treatment of breast cancer. And as you can imagine, I think were 3 or 4 large randomized trials done. There are different companies that market CDK4/6 inhibitors. And there was a tremendous amount of preclinical work done on this, but the preclinical work that was done, and there was a large validation study that they attempted to do to identify biomarkers in the Paloma trial. And there was a lab that was completely dedicated to doing this. Anyway, the interesting thing is the way they classified their Palbo sensitive and resistant cell lines, like the assays that we've used and developed, there is actually very little correlation between the way they ranked their sensitive versus resistant lines versus the way we've done it. We've used a number of different techniques, and Maddie spent hours and hours doing this. But it highlights the importance of really setting stringent standards for the preclinical work because if we don't have stringent standards for the preclinical work, then we get a negative result, as they did in the biomarker study for the Paloma trial. So we're quite excited because we think that we've got a really robust way of classifying the lines, and that we can use this for hopefully to develop a biomarker that works in clinical practice. So I'll turn it over to Marta now, who's going to talk a little bit about the work we're doing in MEK inhibitors in low-grade serous cancer.
Hi, everyone. Yeah, continuing with the discussion of on drug-resistant biomarkers and the need for drug treatment combinations. Today, I'll be presenting a work that was done in collaboration with a lab in the Netherlands, in the Netherlands Cancer Institute, led by Dr. René Bernars and Dr. Catherine Burns, and our lab. And it aims to improve the treatment of MEK inhibitor therapy in low-grade serous carcinomas. Just as a brief introduction, over the last 10 years, our laboratory has been studying low-grade serous carcinomas, and using in-house patient-derived cell line models, we started comparing the different potency of the MEK inhibitors that are available in the market and being tested in trials, which led us to the identification of trametinib as the more potent MEK inhibitor at that time. Doing this type of preclinical testing, we were also able to identify different cell lines with different degrees of MEK inhibitor sensitivity or resistance, which led to the identification of a panel of biomarkers that had potential predictive value in this setting. So this has actually been confirmed by the results of a clinical trial published this year, the GOG 281, in which they tested trametinib versus standard of care in patients with recurrent low-grade serous carcinoma, and they confirmed that trametinib actually works better than the standard of care, that's either chemo or anti-hormones in this group of patients. Response rates went from 4% for anti-hormones and 9%, 4% for chemo, 9% anti-hormones, up to 26% for trametinib. So because of these results were better than the standard of care, trametinib has been now considered the new standard of care for the advanced or recurrent low-grade serous carcinoma patients. So while this is exciting because this is again, it's a cancer that's rare and understudied and it's lacking of effective treatments, we have to acknowledge that there is still a large number of patients, up to 25%, that do not benefit from these therapies. Not only that, the patients that benefit, they also experience considerable side effects, and some of them will eventually develop acquired resistance. So there is still a clinical gap that's important to address. That's how we can improve MEK inhibitor therapy if this is the standard of care. And to do so, we aim to identify targets that are lethal in combination with MEK inhibitor, ideally at low doses to avoid unnecessary toxicities. And we use CRISPR-screened patient-derived cell lines and mice xenograft models to address this question. So just as a brief overview of what we did on this study, we used CRISPR-Cas9 genome-wide library screens to identify genes that modulate MEK inhibitor response in a panel of low-grade serous carcinoma cell lines, ascites, and IgG lines that were derived from acquired resistance, were derived to assess acquired resistance and response to MEK inhibitor. So we use three types of assays: a gain-of-function CRISPR assays, in which we overexpress genes to see if what was the effect on MEK inhibitor response. We also use loss-of-function CRISPR screens to knock down genes and identify those that modulate MEK inhibitor response. And then we also did synthetic lethal drug screens, synthetic lethal CRISPR screens, in which we knock down genes in combination with MEK inhibitor to see which ones would potentiate the drug lethality effects of MEK inhibitor. So all these type of approaches were done in different lines. And all of them pointed to the same pathway or the same gene that was HES1. HES1 was upregulated in resistance. MALM2 and MAP3K1 were two of the candidates that were shown to modulate MEK resistance when altered. So upregulation of MALM2 and mutation of MAP3 resulted in increased resistance. But it was SHOC2 that also regulates HES1 that seemed to potentiate MEK tract lethality when it was knocked down. So because of that, we need to know that there are no current drug or small inhibitors that target SHOC2. SHOC2 is interestingly a scaffold protein, so it doesn't have like a kinase function or any other type of function that could be targeted with a drug. But it has an interesting role as a scaffold protein as it links RAS to MAP kinase. So as you can see in the right diagram, RAS is above SHOC2 and SHOC2 is above MAP kinase. So there was because of that, there is a rationale to use RAF inhibitors in the absence of SHOC2 inhibitors to combine with MEK inhibitors and see if this combination is any better than single trametinib alone. So while we were developing this project, there were two publications that came out in 2018 that proved the same thing that we're trying to prove, that SHOC2 actually regulates RAF function, and it works better when it's combined with MEK inhibitor in the RAS mutant cancers. So what we did next is, once we identified Pan-RAF inhibitors as an alternative for SHOC2 inhibition, we combined Pan-RAF with MEK in vitro models and in vivo models. We use different parental loaded cell lines, patient-derived cell lines. We also use some isogenic lines that display acquired resistance to MEK inhibition. And then we also use some primary ascites cultures, as well as patient-derived xenografts. And in all of them, we saw synergistic effects of the drug combination when using lower drug doses. So at this point, to summarize, the results of this study were that we identified some candidates that modulate MEK resistance, in particular SHOC2 increased MEK inhibitor efficacy. And in the absence of SHOC2 inhibitor, we were able to combine Pan-RAF inhibitors with MEK inhibitors to improve the efficacy of this treatment while reducing potentially side effects, because we are using the drugs at lower doses, and also unnecessary treatment-associated costs. So the next step would be ideally a clinical validation. But as per last year as well, there was there is a new drug that's a dual RAF MEK inhibitor that was developed by a company, and it actually got approval to be tested in recurrent low-risk serous ovarian cancer patients in clinical trials with a combination of a PAK inhibitor. So we're kind of excited to see that the lab results are kind of aligning with some of the trial proposals that are coming out now for this understudied and aggressive low-rate serous ovarian cancers. And we are just looking forward to see our results in the lab can be translated to the clinical side as well. Just to wrap up, we would like to thank all the Carey lab. There are several members to be acknowledged, like Maddie that presented today, Hannah Kim, Sasha Hamilton, Lily McLeland, Josh Yan, Annabelle Sheng, who just joined our lab, Nelson, myself, Dr. Carey, and a bunch of collaborators from Vancouver, from across Canada, from the international consortium, as well as all the funding agencies and patient and partners that have contributed to this work. Thanks all for your attention, and yes, we'd like to welcome any questions or comments